Hi, everyone who just come in. We will start in a minute. Let's make sure that everyone who was in the meeting room a minute ago can get in. Oh, hi, Samia. So it seems like we have like registrants from not just Africa, but from the other part of the world as well. I see Samir. He said he's from India.
It's not good time for India. It's very late, I guess, right?
Late, yes. Yeah, hopefully you have enough sleep.
Thank you, everybody.
All right. Just for the interest of time, I'll introduce Professor Tae Seok Moon real quick. Oh, it's not too bad. It's 9 p.m. In India. Right, let me share my screen real quick. All right. Hopefully it gets up there. Hi everyone. Welcome to the SynBio4ALL Africa Initiative monthly seminar. So we are doing this seminar every we're trying to do it every month, but this month is very special because like we have Professor Tae Seok Moon here from JCVI, and Tae Seok is actually the chair of SynBYSS, which is the young speaker seminar surrounding the SynBio area, where he invited like not just the young speaker, but also the pioneer in the SynBio to talk about their research as well. Tae Seok is also part of the EBRC Engineering Biology Research Consortium Council, where I think that's the first time I meet Tae Seok, where he's providing a lot of insights about the field. And today we are very gratitude to have Tae Seok be with us today, where Tae Seok will talk about his research in the topic of system and synthetic biology, constructing smart and programmable microbes and microbiota to address global problems. So we'll start in a few minutes, but like just to give you a very brief background about Tae Seok. Tae Seok initially came from South Korea. And he went to MIT for his PhD, and then moved briefly to UCSF for the postdoc, but ended up going back to MIT because his whole team moved back to MIT as well. Tae Seok like did PhD under Professor Chris Prather, and trained during his postdoctoral training with Professor Chris White, and he went to be a professor, tenure track professor at Washington University in St. Louis. And now he's just moved to JCVI, J. Craig Venture Institute, and his team is growing. A few of his team members are also like originally from Africa. So he understands your background, like how students from African or like underrepresented background are trying and like thriving in this environment, especially in the SynBio area. So he received a lot of awards, and I think I can't really name all of them. He also served as like a lot of editing roles in the scientific publishing area, and he has a lot of like ongoing grants that we will see a lot of great work coming from his research very soon. So I think like that's a a lot of intro that I'm trying to cover in the short time. But we again thank you a lot, Tae Seok, for like being with us today. And we'll be very thrilled to hear what Tae Seok is gonna introduce us today in the SynBio research. All right, I'll stop sharing the screen now to share to Tae Seok.
So thank you, guys. Thank you so much, everybody, for joining. I know, I mean, this is not good time, John, for Africa, India, wherever you are. It's my honor to be here. I'm really excited to share my vision and also ideas or some data, but not too much data today. because I want to deliver my idea and vision to you. And, I mentioned I have one student from Africa. He's from Zimbabwe. If you don't know him, his name is Tatenda Chopra, and he endured many difficulties in America, including some, racial discrimination, etc. But he overcame everything, and then guess what? He became world hero by developing the first COVID vaccine at Pfizer, and then I now, enjoy my own advice becoming famous, and then we'll hang out again the next month to watch the baseball game together. So this is kind of my big reward, being a professor, not necessarily the publishing paper, but seeing young people becoming big leader, good leader in the world. That's a big reward for me. And then since I can realize those type of things, I devote my time to young people, and then nurturing young people, and then promoting young people, and then advising young people. So if you have any advice you wanted to get from me, I'm happy to do separate Zoom call or by email, and please let me know. And I also mentioned about Tatenda Chopra is one of my students from Africa. I am very interested in Africa problem since when I was very young. Let me tell you one story about my funny story, I guess. My parents in somehow very creative. You may heard about some teaching remark from your parents. For example, you are adopted from somewhere. That is one of the teaching thing from my parents, also other parents in Korea. Most of the parents in Korea basically telling you, you are adopted from under the bridge. That's a typical Korean thing. So we found you under the bridge, and then we start to raise you. That's a typical, teaching thing. My parents is very creative in somehow. They said, we found you from Tanzania. That's one of the African country. I have no idea when I was young, when I heard Tanzania. Just imagine, when I was young, this is almost 45 years ago, I believe. At that time, there's no internet. So there's no way to look at what Tanzania means. And so I have no idea for a while. And then I grew up, of course. And then one point, I believe elementary school age, I wanted to know what Tanzania means. So I alone went to small library in the town, and then I looked it up what Tanzania means, and found Tanzania. That's one of those country in Africa. And since then, I really wanted to do something, or finding my real mom in Tanzania, right? So the reason, they teach me like that is I basically play outside every day, until 8 p.m., just soccer, whatever. So my skin color was a little bit darker than now. So that's why they teaching me that way. So, but now I'm very interested in African problem. I'm really glad I'm now connect with you. So I'm really happy and honored to be here to give up my seminar. That's my intro. So let me share my screen. If I a lot to do. Can you see my screen?
Yes, it's pretty
Yes.
clear, but it's not in the
Good.
presenter mode yet. Now it's in the presenter mode.
Good. So another thing I want to tell you is, I'm giving a lot of talk. I'm traveling constantly, and I probably already gave, almost 200 talks last 3 years. I don't want to do monologue, and that this should be interactive. I know, I mean, the virtual format, interactive conversation is very difficult, but what I ask you to do is please stop by, please stop me whenever you don't understand whatever concept, whatever thing, just stop me and then ask me the question by sending message or by speaking up. Is it okay with ICE and other people?
Yes.
Okay,
Yes,
wonderful. So stop anytime, and then I will stop my talk, and then I'll answer your question. So
Yes, that's
as in I okay, good. So as I mentioned, I'm the JCVI professor. That is I will show you where that is in a minute, and also a council member, EBRC, and SynBYSS chair, and I'll probably talk about that on later. And then this is my email address if you are interested in contacting me. So this is my conflict of interest slide. I got $100,000 from Bob Langer, probably the most famous living engineer at this moment, with a lot of company he founded, and then a lot of papers and a lot of patents. And he got all the awards except Nobel Prize because he's so application oriented, I believe. And then also people said he may not get the Nobel Prize because he's too rich with a lot of companies. But this is rumor, I guess. I mean, I believe he deserved whatever award, I believe. He started a program called Langer Prize, basically giving people, one person per year, money, $100,000 to start a company to translate your idea into reality. So if you are interested, I guess, this year's competition already passed the deadline, but you can do it later next year. And then this is a partner with the AICHE, the American Chemical Engineering Society. And then one thing I want to tell you is, I know how difficult something you could achieve in Africa because there's not many resources. You're still hungry, environment is not good, democracy is not stable at this moment. But I can tell you one story about him. When he was the junior faculty in MIT, nobody appreciated his work. And then this is one of the quote: "No chemical engineering department in the world would hire me." That's basically the Bob Langer, the most famous engineer, actually talking about the engineering a long time ago. And then because at that time, when he was young professor, basically biology is not part of the chemical engineering. People only talking about petroleum processing, etc. And then that's why all the professor basically said to him, you should be doing oil and energy type of research, not the bio. But he continued to do so. And I believe he transferred to another department for tenure. And then he came back as a chemical engineering, now institute professor. That means the highest honor at MIT. And then I believe, he overcame another challenges. So again, I talking about two different things. You probably have much more challenges than any other people because your environment is not good, but don't complain about those things. My student Tatenda overcame huge problem. He's from Africa, from your neighbor country in Zimbabwe. He unfortunately, didn't have electricity when he was young until recently. No electricity at home. His two brother died, of course, because of disease or hunger. His mom suffering from cancer, but still he saved his salary, sending money to his family. And then because he was determined to to make his dream come true. And then he now helping a lot of other people, including people in Africa. He's the role model of me. He's the role model of many people. So you can do it because he can do it. And then Bob Langer also can do that. So this is one thing I want to tell you. So let me go to the real scientific part. So let me introduce synthetic biology in my way. I see the one chat. There might be question. No, not question. No? Okay, no. Okay, so let me start with my concept regarding the synthetic biology or engineering biology. As you know, our ancestors dream of flying in the sky. So think about something you dream of, and then people dream something, and then that became true and reality. So dream high. That's another point I want to make. Even though that looks impossible, but impossible can be possible in the future. So they wanted to fly. But instead of creating birds or transforming themselves into birds, they realized they can make some part of the birds by analyzing birds, something like wings. And then they could also put the propeller together and then assemble them together to make something, some object that could fly. And that is, we now call it airplane. Now we take for granted about airplane, but 100 years ago, that's not the just normal thing, and even people just imagining or dreaming. My slide not okay, moving. And then I want to do the same thing, rather than creating completely new creature, I want to analyze existing cell, for example, yeast, then making beer, wine, or some other product, and then analyzing that one, and then design so-called synthetic DNA and assemble them together to make engineered cell. That engineered cell can do a lot of job for human, including making biofuels, biochemicals, even some food, biomaterials, all the way to diagnostics, therapeutics, and also pathogen control, and also bioremediation. As you know, we currently under big problem, including pollution, plastic pollution, etc. But we could make bacteria to eat plastic or pollution, pollutant, and then clean up our environment to make our life better. And we call it synthetic biology or engineering biology. So this is my concept of, synthetic biology. And I have too many projects at this moment, and I'm constantly hiring people, postdoc position at this moment. One unfortunate thing is we do not have graduate program in the institute, because this is a research institute, not the education institute, unfortunately. So I only can take the postdoc. So if you already finished your PhD, you could apply now to the website. And then I have the multiple projects, so it doesn't matter what kind of project you are interested in, but the important thing is that you should have the clear expertise to work with me, basically. And then this is the JCVI. Craig Venter is a very famous person who, start this institute, but probably it doesn't matter to you because I'm talking about Pacific Ocean. You can watch from the institute, and that is the basic view, from the institute. Craig Venter is basically the person who did first human genome sequencing by beating all the competitors, including NIH at the USA. And then he also recently published multiple papers regarding synthetic cell, that's a synthetic bacterial cell, And then that is another breakthrough discovery or breakthrough, achievement in terms of engineering. So among all the projects, I have, my first project was something like this. And this is my first question when I start my position at Washington University 12 years ago. The question was, can we combine sensor, that's a biosensor, and then genetic circuit, and actuator. Actuator here means the function, like I'll explain that later, and then in a human-defined manner. If that is possible, we can make bacteria or mammalian cell or plant cell work for us for many of purposes we have, including tumor killing bacteria construction, pathogen killing bacteria construction, all the way to toxic chemical eating bacteria or plastic eating bacteria, etc. To do so, you can make, I can give one example. That's my first project funded by Bill Gates. You know the Bill Gates, the Microsoft, founder. He gave me $100,000 to develop probiotic bacteria that basically sense human disease. That is actually African problem. That's my first project. That is a Tatenda Chopra actually working on detecting parasite in your gut using some biosensor. Once probiotic detect parasite in your gut, the probiotic generating some molecule that potentially kill parasite. So we done all kind of different things. Unfortunately, we didn't get the second phase money. That's $1 million. We could not pursue that project, but I constantly thinking about that project, going back to that problem, because we now develop sensor for parasite, sensor for circuit that compute whether you have the parasite in your body or not. And then we already developed one toxin that specifically kill parasite only. So this is the bacteria machine that kill parasite once you have the parasite, once the bacteria detect parasite. So this is my concept of programmable cell. Bacteria looks like robot, but doing job for human being. So that is a basic concept I wanted to implement. So since that, that's 12 years ago, I call this project like constructing smart probiotics, basically sense and response. There are two types of things you can do. I see the one chat. Okay, that's not the question. So basically the concept is the probiotic. That means, you may get the yogurt from somewhere that could be donated food, or you can buy from the some shop. The probiotic containing yogurt basically sense your disease or sense your pathogen in your body, and then in response to that pathogen or disease, basically the bacteria produce some color. That means you could basically say, oh, I take my yogurt, but originally in my body, in your gut, in my gut, there are some disease. That's because of that, I'm basically sick right now. That's a diagnostic application. And then one thing I want to do, I'm sorry, if you already have the dinner or before dinner or whatever, the idea I want to do is you take my yogurt that containing probiotic, and then probiotic detecting this kind of disease or pathogen. Once you have those disease, you basically see your poop, but the poop color is like red color because that means you already have some disease in your body. So this diagnostic application. But there are some other method that is more accurate, that is basically taking blood from your body, or you can also do endoscopy, but that's a typically invasive method, but the more accurate. The result coming out probably 2 days later. But the probiotic idea is very quick, within few hours, and that's kind of basic idea. I see another chat. So this one is, sir, that is nice to hear. I'm thinking of synthetic biology in using microbe in purifying the air with the cooling is as well, like some air conditioner device doing it. It can be some bacteria or okay. That's good idea. Basically algae or photosynthetic bacteria also capture CO2, and then in that way we probably solve problem of the climate change or climate crisis. So absolutely, you are doing a great job. So we're going back to my talk. Basically that is the one application of that's a diagnostic. And second application is of course the therapeutic application. So why we stop there? So as I mentioned earlier, once bacteria detect disease, you can make bacteria produce some drugs or some protein that basically kill the pathogen or cure disease. That's the basic idea of the probiotic. It's not going forward again. Okay, so to do so, we need to have sensor first. As you know, we have the pandemic since 2020 and around a few years. And then our lab also shut down during that time. So we could not do another experiment. So we decided to publish one review paper summarizing what kind of sensor available at the time. Unfortunately, the table I'm showing you is actually the sensor available is around 3 or 4 years ago. And unfortunately, we only have around 10 sensors we can use. Some of that related to inflammation in your gut, some of that relate to diabetes, some of them relate to internal bleeding. But I want to tell you one interesting development by Tim New, my colleague at MIT. They actually use probiotic detect blooding in your body. So basically, the main idea is before, if you wanted to detect blood in your gut, the only way you can do is put your or small camera, we call it endoscopy, small camera all the way into your gut, or even from the bottom. That's painful. I done multiple times, and then look at the bloody area by capturing using camera. That's invasive. It's not fun. But what Tim New did in his, I think, science paper, they basically use small capsule that contain electronic device with the bacteria probiotic. The probiotic sense one of the blood molecule, heme, and then that sensing basically result in generation of luminescence. And then that luminescence signal converted into wireless signal. That means your device outside your body basically communicate with your small capsule in your body. So the only thing you need to do is take the capsule, and then capsule go all the way down to your gut. The capsule basically has the probiotic. The probiotic sense those kind of blood in your body, and then communicate basically at the same time simultaneously. So this is very important because you don't need to do painful long time endoscopy, but you can do real time monitoring whether you have the blood in your gut or not, because sometimes blood in your gut is very dangerous situation. So this is the one thing I want to basically tell you, giving one example. So going back to my study, and then I realized there are too many other, there are only a handful of the sensor available. So the first thing I need to do is developing sensor. So let me ask you a question because I have a question from ICE.
Yeah, so just wanna disrupt with question a bit on the previous one. So like you mentioned a lot of biosensors out there that the biological system can detect sort of, but like how far are we in terms of like telling how much of the metabolites or like those compound are we detecting? Like is it a yes and no? Or is this like can we grade it similar to how we detect like the amount of light or something like that? Just
That's
one.
a wonderful question. So if we are talking about their, analysis using traditional method like chromatography, mass spec, etc., people call it the metabolomics. We already know how much, metabolite we have, especially from some sample from human, but mostly from the mice sample. We have the tons of data, what kind of metabolite we have and how much we have. But in terms of the, sensing using biosensor, as I mentioned, this is the sensor we had 3 years ago. Actually, we did another paper that's a more comprehensive review paper published in, I think, last year. We have the more sensor, but still the number of sensor is handful, like, I don't know, dozens only. So at this moment, this sensor not giving you the real numbers. It can give you a little, high, middle, etc., but not quantitative unless you have the real traditional method to measure and then correlate these two numbers together. Until you do so, we don't know how much we have exactly, but we can do that using traditional method. But biosensor doesn't give that type of number. Yeah. That answer your question?
Of course, yes.
Good.
Thank you.
So because we realize we don't have many sensor we can use for the whatever method, we actually look at some sensor we can develop. So the sensor we tried to focus on at the time was the actually urochemical sensor. I want to ask you question. How many of you heard about neurochemicals found? Okay, let me ask the different question. So you heard about probably neurochemical. If not, thinking about whether you heard about serotonin, dopamine. How many of you heard about dopamine and serotonin? Raise your hand. I see some hand. So I will give some example. So serotonin is, okay, I see some hand. Thank you. Serotonin is the neurochemical that basically control your happiness or your mood. Okay? So very important neurochemical, very important chemical. My second question is, basically you can choose either one or second. The question is serotonin produced in brain or serotonin produced in gut. So you could choose either. How many of you think serotonin produced in your brain? Raise your hand. Okay. How many of you think serotonin produced by the gut cell? Raise your hand. Okay. So if the answer is the brain, I probably didn't ask that question, right? Because the answer is, in fact, serotonin produced by your gut cell. Okay? And then in fact, more than 90%, 90% produced by your gut cell. And then that's a very, very interesting thing I just heard several years ago. Another thing I want to tell you is your human cell, total number of your human cell is actually much fewer than your bacterial cell you're carrying every single day. That means you think you are human, right? But you are defined by your total number of your human cell plus much more number of bacterial cells that basically define your cell. Let me tell you why I'm saying that one. There are multiple research happening last decade. Basically, some people actually one of my colleague, actually my previous university, Jeff Gordon, did very fancy creative experiment. One of them is basically something like this. You have the two mice. One of them has the both of them basically germ free. There's no bacteria in the body. And then they extract bacteria from fat person. Another one is they extract bacteria from slim person. And then they basically put those bacteria into their gut, the mice gut. And then, interestingly, these mice actually twin. But the mouse, which get the gut microbiota or bacteria from the fat person, became fat. The other one getting from person bacteria stay slim. So this is fascinating. So you sometimes, Western people, because they eat too much, they become too big or too fat, and then they worry about. Sometimes they blame themselves, they eating too much, but sometimes, you can say your microbiota or bacteria in your gut potentially impact something. Another fascinating finding was your depression, your mood. Those things actually also affected by the bacteria in your gut. So you are unhappy now, somehow. That's not necessarily you doing something wrong or something bad. Your bacteria, some doing horrible thing for you, because bacteria should be blamed in some time. That's a kind of very surprising discovery that made several years ago, and then I realized I want to study and develop some sensor for the neurochemical. That makes sense? And then I see some question in the chat. Let me go to that one. Let's say yes. Okay, I have the one question from Emma. Are these sensors on market or they are still under research? Most of the sensor is actually under research right now, because this is a pretty new field. And then second question is there an easy way SynBio? Let me see, SynBio. I have some interesting. How can I get rid of this guy? Make it bigger. Okay, now see the better. And then is there any easy way SynBio can help to develop this technology in Africa apart from education, which we are currently offering? So a sensor have been developed by a lot of young people, including iGEM competition teams. So the answer is yes. If you are passionate about solving the problem, that is the problem, especially I guess the problem in Africa. For example, malaria drug. There are malaria drugs, but you can make the malaria sensor. There are many problems in Africa, including water quality. But one of the problem is some heavy metal that kind of very toxic. You can develop heavy metal sensor to cheaply detect heavy metal using bacteria. There are multiple different ways you can do. There are multiple examples also developed by the African IGEM team. So answer is absolutely yes. But you need to be persistent, work hard to develop those things. Actually, I helped some of the IGEM team in Africa to achieve those kind of goals. And then they found a company based on their IGEM competition project. And then this is the basic answer, brain, gut. Interesting. Gut cell and only about 10% in brain. Yes, that is the correct produced in the gut. This is the basic answer. And then the question is, are probiotics only microorganisms? There are multiple, again, I'm not sure though what is question. Probiotic, typically microorganism. Some of that is yeast strain, Saccharomyces boulardii, and then but most of other probiotics are bacteria like E. coli Neisseria or some lactic acid bacteria, etc. And then the Emma said I actually mentored IGEM team last year. Okay, the biosensor. Good. I believe I covered all the questions so far. Any other question before I'm moving on? Okay, I don't see any question. Okay, I'm moving on. So I realized I need to develop sensor. So one of the sensor I want to focus on is neurochemical, as I mentioned. The reason is something like this. For example, serotonin. Serotonin, as I mentioned, produced by your gut cell. And then that means many of the serotonin actually found in your gut. And then that influence your happiness. And of course, in this era, there are too many problems outside, including war in Ukraine, war in Israel, war, there are many problems in Africa, climate crisis. This is a gloomy era. Of course, every single time, 10 years ago, 100 years ago, very gloomy thing, happening. I personally had huge problem because of discrimination and harassment and retaliation against young people, people of color, including Tatenda Chopra in the university. So we are living in a gloomy world, unfortunately, because people like me, old guy, screw up because we need to give you better world to young people, but we didn't do that job. So this is unhappy situation, environment. How we gonna be happy? That's a question. So to make you happy by using scientific effort, I wanted to develop, for example, serotonin sensor. The idea is basically using bacteria, and then that sense serotonin. And then you take my yogurt, and then you look at your poop. And then let's say if the serotonin level is 20 to 100, let's say that's the right good level. Because too much serotonin is not good, too little is not good. So let's say 20 to 100 is good level for serotonin. And then bacteria produce pigment, as I mentioned earlier. If the level is too low or level is too high. And then if that is the case, your poop color become red. You understand so far? And then that means today is the unhappy day. Don't do anything. Stay at home. Just do not do any stupid thing. But life is sometimes you have something happy. So if that is the case, your serotonin level probably 20 to 100. and then your bacteria now produce green pigment, and then your food color become green, and then you are happy day. Today, you do whatever you want to do, and then because you are happy. I'm sorry. But why we stop there? So one thing I want to do using this sensor is you basically engineer bacteria. And one piece of data I didn't mention is there are multiple bacteria basically synthesize serotonin, also degrade serotonin. So if you kind of connect your sensor, and then in response you're sensing about serotonin, if your level is too high, you can make bacteria produce enzyme that degrade serotonin. In that way, your serotonin originally become too high, more 100, but enzyme that degrade the serotonin and the serotonin level become lower. If your serotonin level bacteria detected is too low, bacteria start to produce enzyme that synthesize serotonin, and then your serotonin level going up, and then you basically maintain serotonin level like 20 to 100 all the time. This is something like we call it homeostasis or dynamic control, and that's sometimes people maintain the temperature in the air-conditioned room at the certain temperature, that set temperature, basically. So I want to do that. And then based on that idea, I already found a company called Moonshot Bio. And that's actually I already introduced earlier with the $100,000 from Bob Renger. And then I already have the punchline for my commercial, even though I still need to develop lots of things to go there. And then my punchline in commercial is basically something like this. Do you want to be happy? Take out my yogurt and you will be always happy. So that's my dream, to make you happy in the future. But as I mentioned,
Sir, I have a doubt.
Somebody have question?
Yes, sir. Sir, Samir here.
Yeah, please ask question.
Sir, my doubt is that will it not increase the product abuse or the microbial abuse in the people because they will be getting some high or something like that with your serotonin product or yogurt. So it will not be conducting some microbial or some product abuse like that.
So can you repeat that question? I actually didn't understand what is your real question.
So basically, I'm trying to say there are some kind of products now, like people used to abuse them. So it can also be like that only because it will increase the neurological systems and all things related to that. No, people will be getting happy and everything. So getting high serotonin and everything like that.
Oh, I see. So you worry about abusing some drugs.
Yes, sir. Yes, sir. Like that.
Yes, absolutely. So I'm giving you the example of serotonin to make you pay attention to, but I'm not actually developing serotonin as my potential target. Actually, I actually, yes. Let me give my perspective about happiness. Happiness coming from your mind, not from the drugs or chemical or food. Happiness, I believe, coming from your mindset, because I know now, because I suffering from my literally dying years. I literally dying for 4 years because of discrimination originally, and then misdiagnosis. And I suffer from those disease for 4 years. I went to emergency room multiple times. The only thing the doctor can do at the time was giving me morphine, the painkiller, but that's also drugs, the bad drug. But that is the only thing that they can do to make my pain go away. But now I do believe, after my miraculous recovery, if you think positively, you could maintain happy all the time because I'm actually happy. I'm also actually happiest ever in my entire life. That is actually start 2021 when I changed my mindset. Let's think positively. And then that's the point actually my Tenda Chopra actually wake me up, teaching me life is not about money. Life is not about reputation. Life is all about you think positively, you think you could do something to contribute to the society. And then my mission I found, probably given by the God, and then my mission is helping young people like you. And then I am happy even though I only sleep 4 hours a day, because I'm very happy I cannot sleep because I have too many things to do before I die. So my personal opinion about happiness is change your mind, be positive, even though you have a lot of problems. I know there are, as I mentioned earlier, world is not the world you want it to be, but you can change it if you're passionate about, and that's my answer to happiness. My real goal in terms of this sensor project is about the actually first part, that is a phenylketonuria. So if I answer that question, more extensively, phenylketonuria is the disease some newborn baby develop because they don't have the enzyme or metabolism that degrade or utilize phenylalanine. The problem of phenylalanine accumulation is sometimes newborn baby die. So typically currently, the way to do is, especially the developed country, they screen when baby was born, and then basically screen whether they have the enough whatever metabolism or gene to metabolize phenylalanine. And then if they become if they found they have the PKU, phenylketonuria, the only solution or a few solutions we have currently is, one of the main solution is diet restriction. That's the very painful because phenylalanine is everywhere, especially in the meat. And then protein should have phenylalanine. That means you should restrict protein food. It's almost impossible, right? So most of the young baby with that disease cannot grow well because they should restrict phenylalanine consumption, and then that's kind of not ideal. So some company already developed some enzyme, but that's an injection by needle, and baby doesn't like that treatment. So because of that problem, some company like Synlogic recently developed phenylketonuria treatment using probiotic. And unfortunately, recently they stopped doing so because this is a rare disease. And then probably there are multiple problems, including economics problem. I don't know exact reason, but they show the efficacy degrading phenylalanine to low level. But again, the problem is too low is bad because baby cannot grow. So my idea is basically using the same concept, serotonin level the same range, I can make the phenylalanine level same range in an ideal level. That's what I wanted to do, in fact. But I want to give the fancy, exciting example because everyone wanted to be happy. So, but that's not the project I want to pursue. That answer your question?
Yes, sir, surely. So that thing explained got me two questions more, which is basically one is that what will be your product or something like that, as you said you're having some company and all. And another thing is that who will be your target audience, where you will embed this market and something like that.
So what do you mean by the target audience? What is the target consumer?
Yes, sir, target consumer.
So for the phenylketonuria, of course, target consumer is, of course, the patient. So if we have the probiotic that control phenylalanine level in whatever level, they don't need to worry about phenylalanine level. So that means they can eat protein. that containing phenylalanine. If you're talking about neurochemical, about serotonin, the target consumer is everybody. But I'm not pursuing that thing because I believe, again, happiness coming from your mindset, not from the drugs or chemicals. That answer your question?
Okay, sir. Understood. Understood.
Okay, good. Any other question? That's good.
Yes, sir.
Any other question from other people? I start to enjoy my talk because I have no more question now. Let me check whether we have question from audience. Do you have any question from chat?
Yeah, we do have a few questions in the chat, Tae Seok.
Okay, let me go. Okay, how can I go that one? I lost my chat box. Oh, I found it. Okay. Okay, so where should I go? Go down, down, down. Oh, okay. Is serotonin exclusively produced in the gut? Serotonin produced more than 90% of serotonin produced in the gut cell. Yes. Serotonin can be produced in the brain. Brain is very small amount, less than 10%. Yes, less than 10%. And then addiction, we already talked about, I believe. What about cortisol? If its level is also high, I mean, cortisol level should be also in certain range, not the too high is bad, too little is bad. Serotonin can also be produced in the lining of intestine, which control appetite. Yes, absolutely. That's what I'm talking about. More than 90% produced in the gut cell. Are you talking about this source of serotonin? Yes. So basically, the serotonin produced mostly in the gut cell, gut lining, intestine, etc. Yes. That answer questions, I guess. Okay. Let's away from the serotonin because that's not the research I'm talking about. So the real problem of sensor development is we have some sensor in biological system in nature. The problem is the compound we're talking about is very similar, and then most of the sensor is not differentiating between the small change, small difference chemical, like I'm giving some example here. So one example is here in this slide, PEA, phenylethylamine. That's a basically compound with amine and aromatic ring. The other is actually tyramine, another neurochemical, but the only difference between these two guys is one oxygen or no oxygen. The problem of sensor that not differentiating between these two is huge. If your doctor has sensor, but that cannot tell difference between these two compounds, you somehow go to the hospital because of food poisoning, but your doctor may say you are crazy because you have the psychological disorder. But that's completely, misdiagnosis. Those kind of things happen to me because they didn't pay attention to what I described when I was sick, but they just randomly talk about something and then prescribe some drug that basically giving me side effect that make my life miserable for 4 years. So we need to develop very specific sensor, only detect one, but not the other. But I realized I already spent 1 hour, so I will speed up because I need to cover many things. Let me go next. So we try to reproduce all the literature sensor data by repeating the same example, same experiment. Unfortunately, we spent 2 years. Actually, Tatenda also spent some years, but we couldn't find any sensor working for us to detect neurochemical, including PEA, TIRA, dopamine, and tryptamine. That's all neurochemical, structurally very similar. One oxygen, no oxygen, 2 oxygen, etc. So we stopped, and then COVID just hit us. But before we got COVID and then shut down the lab, we realized we should do something completely different. So generally, sensor development done like this. You have the small chemical you wanted to detect. In this case, this white the circle. In this case, aldehyde. Aldehyde basically bind to the protein, this the square, and then that binding basically change the protein structure, conformation, and then that basically allow this complex bind to the DNA, and then DNA basically express, and then you now have the GFP or pigment enzyme, and then that produce color or fluorescence. We found this one after 2 years of struggling, and then we basically realized this is not the real amine sensor, but this is actually aldehyde sensor based on our experiment without and with enzyme. Fortunately, when we claim this is a really amine sensor based on reaction rate, TNA is very, very fast. It's within a few seconds or the minute maximum. And then this aldehyde is unstable. So this one actually bind to this one, but this is also converted into the carboxylic acid. And then because of that, this aldehyde not found in diet, not found in the gut. So we can say this is a really amine sensor, and then reviewers of the paper actually buy that argument, and then we actually publish the paper in Cell System. And then, but still there are multiple problems at the time. When we found this mechanism, we call it two-step. This is not ideal but this is a fancy word of the dual non-sensing mechanism. Two steps still take longer, even though this is a quick reaction. So we need to develop much quicker, but the main problem was all the 4 compounds actually detected with some different degree, but that is not specific at all. And then we got COVID and the lab shut down. And so we couldn't do any experiment for several months. And then one day, actually one of my another student, Matt Amrofel, I believe the ICE know him very well. He's the president, used to be president of the EBRC, Student Association. He realized he need to graduate soon, but he cannot do any experiment, and then he wanted to do something computational thing. So he sent me email one day, professor, I wanted to learn program that is developed by the Dave Baker and some other people that's used to be ICE institute. I want to learn Rosetta and AlphaFold. That's basically protein structure prediction algorithm. To me, nothing to lose. So I encourage him to work on that one to learn program. Several weeks later, without my feedback, he mastered how to use AlphaFold and Rosetta. And then he said, I know what to do to predict. And then I'm so delighted. Without my input, he independently mastered program. And then I asked him to, okay, go forward to predict what kind of residue we need to change to make sensor specific. He come up with the 5 different residue after doing docking simulation. And then we found this is much easier because before we did so-called directed evolution that required generation of library up to 10 to the 7th variant of protein or 10 to the 8th. There's a lot of, protein variant, but we screened all of that, but unfortunately we didn't get any good hit because of false positive or false negative, or a cheater kind of sneaking in, giving some wrong signal in terms of that screening. But now we only have 5 residue. We can easily change one residue to the other 19. So we can screen 20 each and total 100. And making 100 different plasmid is nothing to us because we are making all the plasmid every day. We can make a couple of weeks or 100, and then we screen all of that. And then we found many specific sensor. And then one of them, actually, I'm showing here, there's a PRA change into the L amino acid. And then we find very specific sensor that is only detecting PEA, but not others. So this is the basically one snapshot of the research. I'm kind of taking too long to just 1 hour to talk about, but in summary, this is a first specific sensor for neurochemical. And then we even didn't have crystal structure of this protein, but we now giving some insight about this sensor. I believe this is a generalizable to develop some other sensor, in the world. I see one more question from audience. Could you please share your insight on the potential and challenge of using synthetic aptamer and their application? Absolutely, aptamer is a great avenue you can explore, and there are many different ways to improve and then engineer aptamers, including Select. That's a continuous kind of evolution method developed by multiple people, but including some Andy Ellington and others. So you can use that one for sure. Second question is, so I am not able to understand that what is your product is some dietary enzyme, which is the detecting serotonin. Serotonin is the example I just giving you some exciting one. We are not developing serotonin sensor. We developing sensor for some other neurochemical. Now we developing dopamine sensor. We almost done, but we developed so far is a phenyl PEA. I already show you here. I'll go back to the slide. So we basically now have the sensor for PA. This is one of the neurochemical. The impact of PA is some related to psychological disorder, and then tyramine that related to food poisoning. But this is also some blood pressure increase related neurochemical. So we are developing those things. And then go back to question chat. So yeah, we are not, so but PKU also the one of the digit we are looking for. PKU and serotonin nothing to do with there's no relationship between the PKU and serotonin. Did I answer the question? Okay, I guess that's answered. Any question about this first part?
Hi, Tae Seok. That's very cool. I have one quick naive question to add here. So you mentioned that Matt and also the team are like thinking about using more computational protein design into the process of like designing this sensor. But as far as I heard from the team, let's say like several years ago from the Vega Lab, it's not trivial to design the protein that have multiple states, because like when we try to make them allosteric, like the current state of the art is not up to the the level that we want yet. So is there any other tricks that you are like using that along the way? Because like trying to design one thing or design one compound that can behave in two states are like very different.
Yeah, so that's absolutely great question. So let me give my perspective about the protein engineering in general. So when I was a graduate student, we use state-of-the-art technology at that time, molecular dynamic simulation, hardcore molecular simulation using protein, simulation. With some people from the Dean Wittrop lab and those people, the very high level, the expert. He gave me 20,000 variant, or actually 10,000 variant I need to screen, even after prediction. but that's what I'm talking about 15 years ago. And I became a robot, picking colony more than 20 because you need to cover, if you want to cover 10,000 colony in the variant, you need to cover, you pick the probably 30 or 40 different thousand colony. So I basically use toothpick, become robot myself, pick the colony, pipetting more than 100,000 because we kind of use the triplicate experiment, and we didn't have liquid handler at the time. I was so unhappy, of course. This is some labor intensive. I promised to myself, I'm not going to do protein engineering in my life anymore. That was my promise. And then I start protein engineering project because we have now liquid handler because I don't want to make my student doing labor intensive work. But we rely on the directed evolution, but it didn't work. I also need to use directed evolution because my academic grandma is Frances Arnold. I should use her method, and it didn't work. It worked for the some method, some protein, but it did not work for my specific sensor. So going back to your question, there are still many challenges we need to overcome because the protein prediction, structure prediction, docking simulation is not perfect. But thinking about my 15 years ago, my PhD period, and then thinking about now, tremendous improvement. And I'm now seeing the prediction become more accurate. And then for my case, I was surprised. The docking simulation giving very nice 5 different residue, and then changing that on, giving very interesting specific sensor. And I didn't expect that one, but I just want to do, and then Matt deliver beautifully. So another thing I want to tell you is the only contribution to the research is something like this. I don't have time to do real experiment myself. I just sometimes stop by my lab saying, how are you doing? And that's my real contribution, asking the question. And then people normally said, oh, professor, nothing worked for 3 months. I don't know what to do. I normally do not give answer. I just ask, what do you want to do? Because they already suffer from 3 months of failure, right? They have the multiple different idea, and then they give me several options to do. And then I just encourage them to do A, B, C, D, E, if that's the feasible and reasonable. And that's the only my contribution. And then 3 months later, I came back and then say same thing. How are you doing? And then 3 months later, they kind of said, oh, professor, by the way, now one thing working. My real contribution to the project, patting their shoulder, saying great job. And that's it. And then 3 months later, they put my name as an author. And then people remember me rather than fourth author, unfortunately. So my point is, my job is not necessarily knowing everything, teaching everything to the young people. My job is inspiring and then giving direction, high-level direction, vision. And that's why I'm not presenting today data-heavy talk, but I'm giving some story and ideas, vision. And that's what I wanted to do because that's typically I'm doing for my student. And then Matt and Rafael beautifully deliver beyond my expectation. And then I actually really appreciate it, respect those kind of young people because they deliver, they really solve the problem. I'm basically helping those people. So I believe in the future, this prediction tool, AI, computational algorithm will overcome all the problems you just described because people like you, young people, working hard every day to improve those kind of problems for people. That answer question?
Yeah, that sounds good.
Wonderful. So this is the paper I talking about earlier. We now have extensive review paper. Probably longest paper I written, 20 page, more than 200 citation. Chen Gang did great job summarizing current state of art. So if you are interested in sensor development, this paper might be your textbook for now, I guess. All right. I already spent too much time, so I probably need to go quicker. So the problem, I kind of already, present so far is basically about making GMO. That's a genetically modified organism. I'm not sure the situation in Africa, but GMO, people hate GMO. And then I'm making a lot of GMOs. And then I had the one experience with the one former ex-news anchors debating about GMOs. And then at the end of discussion 10 years ago, he called me, "You are a bad guy because you making GMOs." And I got it because I'm making a lot of GMOs. Since that, I'm thinking about what we can do, make GMO as safe as possible, because we don't know what happened with GMO. Because you may watch too many movies, bacteria become monster, even dinosaur, I don't know. So that's my goal. So this is coming from the one movie clip, Mission Impossible. I gave the mission to the bacteria, solving the problem, including detecting disease in your gut or curing the disease in your gut or curing PKU or whatever. Once that job is done, this bacteria, live bacteria, coming out of your body. And then I need to make sure that bacteria not surviving in the environment, because we don't know what's going to be happening. So that's because of time, I'm kind of probably going quicker. So to do so, we use the Cas9, the CRISPR system. I believe you already heard about Cas9, CRISPR. To remind you of the real fact, Cas9 is not recombination tool. Cas9 is basically seizure that cut DNA specifically in some specific region, that basically tool destroy DNA in that way, using as count selection method. We use the Cas9 CRISPR system as a seizure to destroy DNA to kill bacteria. So that's the main idea. We use a Cas9 because Cas9 actually applicable to many bacteria. And also bacteria typically do not have the non-homologous end-joining mechanism. That means even if you cut the DNA, there's no way to stitching them together. That means in theory, bacteria should die. Unfortunately, no one want to die. No bacteria want to die. So the problem of this one is, this is the Austin's project. He's the amazing guy. He spent 6 years to make this work, published. And that this is, I believe, after a few years of struggling, failing all the time, and I finally got this one figure after several years. I will explain that one because this is an amazing, good one figure he generated after several years. So this is basically the data. Y-axis is the colony forming unit, number of cells in essence per volume. And the first data set is basically control, the most important thing. In your experiment, the most important thing is the including control. This control is the no guide RNA control, that means no kill switch. Gray bar is the zero inducer, that means it should not do anything. But this is a control without kill switch. So after adding inducer, that total number of cells should be same. So that is actually the I'm showing here. And then the rest of them is basically with the guide RNA. That means the bacteria die, a lot of bacteria actually die from 10 to the 8 to 10 to the 4. You got it? So that's a lot of killing. This is a massacre, basically killing a lot of bacteria. So all the bacteria hate me because I kill a lot of bacteria. The question I want to ask is, let's say we start with the 10 to the 11 cell per ml. To give some idea, if you done some experiment before, OD of 1 of E. coli, typically 10 to the 9 cell per ml. 10 to the 11 cell means OD of 100 or 200. I'm not sure how many people achieve OD of 100 or 200. That's only achievable using a lot of feeds of glucose, continuously feeding fed batch in large-scale bioreactor, controlled pH, etc. That is the only way you can achieve OD of 200. So let's say we start with the 10 to the 11 cell, and then let's say I achieve killing all of them, except one bacterium. The question is it success or failure? If you think that's success, coming from 10 to the 11 cell all the way to 1 after killing, please raise your hand. No one? Oh, come on. Thank you. So let me give some another idea. So NIH, US NIH guideline is if you start with the 10 to the 8 cell, and then you kill everything for the biocontainment, and then one bacterium remain, they are okay with that one. That's the guideline. I start with 1,000 times higher cell number, and then I kill all of them except one. That means that we meet the NIH guideline. Okay? Let me ask you again. Is it success? Please raise your hand. Oh, nobody except. So failure, raise your hand. Failure. So Samir, you think that's a failure. Why is it failure? No,
So I
no...
raised for success. Success.
Oh, you think of success. And you don't think that's a failure? So actually there are two answers to that question. One is success, the other is a failure. Let me give why in this case. If one survivor exists, that one survivor become 2, because bacteria divide. 2 become 4, 4 become 8, 8 become 16, and then all the way to dominating population, right? So in that sense, this is a failure because you don't know what's happening with that one bacterium. But another answer to that question is, so far for the last 30 years, people study GMO competing with the native bacteria, and then none of the case show GMO survive when it comes to competing. This is something like this. If I give my 2 students, one student giving a lot of homework, the other student giving nothing, and then student without any homework happy, and then physically stronger, and then at the end of the day, not that much tired, and then with homework, the student with homework, they not competing with the other. That's a much happier. Same thing, GMO has additional homework task that's not beneficial for their survivor, but we are giving that additional homework, and that they are not typically surviving well in native environment, And then that's 30 years of the data. But I cannot say that one because a lot of public, watch too many movies, GMO becoming monster. So I need to make sure all the bacteria should die. So unfortunately, my student, Austin, suffered from one problem. That's the probably biggest problem in biology. That's a mutation. He observed many mutations. That is basically one example I'm here. This is basically deletion of the promoter that driving Cas9 expression, and then we do not have functional Cas9 because of this kind of deletion. And this type of thing happening multiple times whenever we do multiple different things, like we kind of knock out deletion of error-prone polymerase that actually exists in the cell, but even if that deletion still giving some mutation, some other place, etc. So we are basically stuck. But one day, as I mentioned earlier, I went to hospital too many times, and then America, if you think America hospital system is good, it's much better than Africa hospital system for sure, but America system-wise is not good because you need to wait 2 hours still, like, and I don't know, from similar to Africa, but only for the 5-minute consultation. So I waiting for my doctor for 2 hours, even though I book or reserve my spot, and then I didn't bring anything, computer or cell phone, so I don't have anything to do except daydreaming. And I asked question to myself, if hospital lose power, can doctor perform operation? The answer for the developed country is yes, because fortunately in developed country, we have the backup generator in all the hospital. If they lose power, the backup generator kick in, and then basically electricity is still going in. Having said that, and then thinking about that situation, I realized why not? We can have backup kill switch that potentially also working if one kill switch not functional, and then we can kind of have the additional kill switch. So it's kind of easy to say, but Austin spent another 2 years to integrate all the kill switch up to 4, and then after integrating all 4 kill switch into the genome, he finally showing the data like this. This is a mouse data, and then basically you see all the control bacteria stay the same number, CFU, colony forming unit, but with the kill switch, bacteria basically die within 48 hours, completely without any survivor. And then this might be, this is using ATC, the one chemical, because we wanted to kill bacteria in the gut if they become pathogenic or become bad guy. But this is not the real containment because we wanted to kill bacteria once bacteria, accomplish their mission in the gut. But once bacteria come out of your body, bacteria basically wanted to, we wanted to kill the bacteria, but we actually use the temperature sensor. And this is a temperature sensor we developed based on the other colleagues in the data. Basically, this temperature sensor sharply transition around 35 Celsius. That means the 30-degree kill switch working, and then 37, that's our body temperature, kill switch not working. That means in your body, bacteria survive and then do the job. Once bacteria come out of your body, and that's typically outside, that is actually around 30 degrees. Even if Africa, I believe at night, sometimes the temperature go down below 30 degrees, even hot summer. That's what I heard from Tatenda. So that means the bacteria basically die at night because temperature go down 30 degrees. We demonstrate using the mouse again. And then we basically kind of developing some other kill switch using some other application like photosynthetic bacteria capture CO2. Some of you actually comment earlier. And then, but typically that photosynthetic bacteria used in outside, but we can also control if the photosynthetic bacteria become too many cells. That's typically bad because they're kind of giving a lot of other problem. But we can add specific chemical like ATC, only killing photosynthetic bacteria, but not others. We can also use that for some other application like plastic eating bacteria. Once they eat all the plastic, they basically die. We recently have the one project almost done. That's basically phenol eating bacteria. Phenol is a very bad chemical that causing a lot of problem, including cancer. But what we develop is cancer causing phenol consumed by the bacteria. Once this bacteria consume all the phenol, they basically die. So probably how many minutes do I have, Ice?
Yeah, I think there is around 20 minutes left.
Okay, so let me speed up. So if you kind of interested in metabolic engineering, you probably already done something like feeding glucose, one bacterium, one bioreactor, one flask, and then making one product. That's a typical metabolic engineering project for last 3 decades or more than 3 decades. But why are we thinking about this one situation rather than thinking bigger? So recently, after 10 years of fomenting the idea, but I didn't want to put that idea into the world because this is already crazy. Probably big name in metabolic engineering, probably not gonna happy with this idea. But I at least did, recently. Now my idea is something like this. Soil, for example, or water, or entire planet can be viewed as a huge bioreactor. In that bioreactor, we have the photosynthetic bacteria capture CO2 to potentially reduce greenhouse gases. Another problem in Africa, huge problem is crop yield because you do not use much fertilizer, nitrogen fertilizer, ammonia. We, Westerner, in developed country, we use too much ammonia that sometimes giving a lot of problem, including algae bloom, because ammonia going into the ocean, a lot of algae bloom that kill all the ecosystem. But we can use some bacteria fix nitrogen. There are native bacteria, but there are some company called Pivot Bio. Also, they are my friend. They actually develop bacteria fix nitrogen. If you use that bacteria in the African crop field, you can fix more nitrogen. Crop yield will increase, and that might be a good way to use metabolic engineering to help agriculture. As I mentioned earlier, we can use plastic eating bacteria, convert microplastic into high-value chemical, generating more value. That's another way to do with this concept. So now I changed my concept bigger, not only one bioreactor, but huge bioreactor in the planet. To do so, we need to have some tools to engineer. So last several decades, we advanced the knowledge and science by using knockout tool. Knockout tool help us understand the function of individual gene in the single cell. But why not? We are developing some tool to understand function of individual strain in the microbiota. In the soil, for example, we can delete one or kill one strain. And then some phenotype of community change. In that way, we can say, oh, this bacterium doing something. If you kill nitrogen fixing bacteria specifically, you now lose nitrogen fixing ability, we can say, oh, this bacteria probably doing fixing nitrogen. We need to isolate this bacterium, for example. So to do so, we developed one method. Again, the Austin did second great work after using the kill switch concept, but in a microbiota, scale. He developed algorithm that's a prediction algorithm using machine learning. That algorithm basically predict what kind of guide RNA we need to design computationally to specifically kill only one bacterium out of billion different bacteria in theory. With that guide RNA, we can deliver that guide RNA and DNA using liposome. Liposome is basically bilayer, lipid bilayer that non-specifically fuse with any bacteria with a whatever membrane, fuse, and then insert that DNA into the cell, and the specificity coming from Cas9 and the killing specific bacterium only using Cas9 mechanism. In that way, you can kill one pathogen in soil specifically without touching, because if you use chemical or antibiotics that kill all of them, but that's not good because you also killing beneficial bacteria like nitrogen fixing bacteria. That's not the good way to go. Also, chemical contaminate soil that also affect human. So that's the one thing we develop. The other way you can do is you can integrate the specific antibiotic gene into the genome. In that way, you can isolate only beneficial bacteria like nitrogen fixing bacteria from the environment, because isolation is a very difficult process, even take 5 years of PhD work for one isolation. And then, but now we can facilitate the process. We demonstrate that it's possible using real world sample. In this case, we use the mouse poop, spike our target bacterium, and then we isolate or only kill the target bacterium using mouse poop. One last thing I probably want to talk about is about the cloning. How many of you done cloning, making plasmid in your life? At least once. Raise your hand. I see some hand coming up. How many of you kill your bacteria after you finish your experiment? Everyone should do that because that's government regulation. If you do not kill bacteria and then release that into environment, you probably end up in jail. So please do kill bacteria after your experiment. How many of you destroy DNA after your experiment? I don't believe you destroy your DNA ever. Did you?
Some of them.
How?
Throw them into the bleach.
Bleach doesn't destroy DNA, that only denature. But once that become outside and dilute, DNA come back double-stranded. Autoclaving doesn't destroy DNA. Autoclaving, again, denature, and then temperature go down, attach together again, and then that become the problem I'm thinking about for 10 years, but nobody done that. How many plasmid we dump into the environment for last 50 years? We dump too many plasmid that containing antibiotic resistant gene. We blame people like farmers because farmer is the #1 user of antibiotics. Probably around 90% they use. Second, people use a lot of antibiotics, of course, in clinic, doctors. We blame those people because whenever we heard about superbug that resist against antibiotics. We blame those people, but should we blame ourselves because we dump for last 50 years plasmid that containing antibiotics and release into the environment? We should blame ourselves. We need to prevent something. But I search literature to understand how many plasmid in the world distributed, but no paper published about that one. So I changed my strategy. Rather than studying the impact of the antibiotic resistant containing plasmid, I want to develop new method that does not contain any antibiotic resistant gene during cloning process. If you think that's too complicated, I can give some high-level idea. You can use auxotrophy. Auxotrophy actually used for selection in yeast, but interestingly not in bacteria. But to maintain the plasmid, you need to have another knockout in the genome. That's an essential gene. Because that essential, as soon as you knock out the bacteria genome, the gene, the bacteria supposed to die, but we have the clear, interesting idea, the maths work again. We have the plasmid, intermediate plasmid containing both auxotrophy gene and essential gene. And then your cloned, Gibson assembled cloning plasmid or whatever plasmid you have, if you do normal cloning transformation, that plasmid going in, and then you have the two plasmid. One is the intermediate, but that intermediate plasmid kick out because this one is temperature sensitive or sucrose sensitive. So only your plasmid remain. If that's still very complicated, you don't need to worry about. You just go to the add gene. You can pay some fee. You can get this plasmid from the add gene. You can now do not feel guilty by contributing to antibiotic resistant gene spread problem by using this new cloning method. Same efficiency almost, and then you don't need to use antibiotics. That means save your chemical cost, but you also not contributing to the antibiotic resistant problem. So this is data, but I skip that on. I believe, I only have, 10 minutes each. This is a metabolic engineering project, but I already kind of mentioned some of the problem. Let me give some idea what I want to do. What I want to do is all the plastic. Currently, we have the technology, including polyethylene terephthalate, PET, polyethylene, polystyrene, polypropylene, mixture of them using chemical method, and then break down into the small molecule. We found bacterium, two bacteria, that converting that into high value chemical. There are multiple different ways to convert from PET to the PET. There's one company from French company called SynBYSS. They're doing amazing job to make the PET. The problem of PET making is the cheap, too cheap. That's only $1 per kilogram. Entire process costs $2 per kilogram, and that's not gonna work economically. The way they working right now is the French government giving a lot of money to the company to make that happen. What I currently developing is not only from PET, but as I mentioned earlier, PET, polyethylene, polystyrene, polypropylene, converting that into small molecule using chemical method. And then you use the biological method, bacteria, two bacteria consuming almost 100%, regardless of the composition of plastic. That's a keyword, because your plastic waste different from my plastic waste. Today's plastic waste composition is different from tomorrow, but our two bacteria, regardless of the composition of plastic, we almost 100% consuming all of the plastic waste, basically converting into the small molecule, and then we now making carotenoid. Carotenoid is something like several $100 per kilogram product. But this is a high price, but I not that much interested in money. I'm interested in more likely the real conceptual problem. Currently, carotenoid produced by company, especially some Chinese company and etc., they use carrot or tomato to extract those high value, high price chemical from the food. I hate that concept, that practice, because many people, including Tatenda's family before, did not have enough food, and 2 brothers basically died. But at the same time, people in another part of the globe using food, extracting high value chemical, that's conceptually not acceptable. So I want to disrupt this market by introducing this technology that's basically using plastic waste all the way to high value chemical. If I deliver that on cheap enough, all the company in China will be completely bankrupt, and then they do not play with food. And that's one thing I wanted to achieve. So we are going that direction. And then actually this is all the data, but bottom line is we start with the plastic, and then we use the chemical method, as I mentioned. And then this is all the data with only PET conversion, but we currently making lycopene up to 1,000 microgram per liter. And then we actually improved that one, but I don't have the data in this slide, but up to 20 milligram per liter we're producing right now. This is not commercially viable yet, but 3 years from now, we can make the gram per liter lycopene, and then probably that is the point I probably retire by selling this technology, and then probably stay mentioned in somewhere, and potentially also start to talk about making synthetic biology university in Africa. I currently discussing with my colleague, so I will probably become president of one university in Africa focusing on synthetic biology in the future, if this one is successful. So I also doing some computational work to design the enzyme that is a mesophilic enzyme that degrade PET at room temperature, but still very challenging. But we use the AlphaFold Rosetta to design, and then we synthesize all the 500 kilobase DNA, partner with the JCVI DOE. And then once we have the mesophilic room temperature functioning enzyme, we can put that enzyme into the mycobacteria, and the mycobacteria secrete the enzyme into the wall, and then degrade PET, and then take the monomer into the cell, and then convert that into high value chemical, etc. In that way, we clean up our environment. Of course, that bacteria should have kill switch. So this is a seminar series, ICE already introduced. So I'm YouTuber. That means you need to subscribe and then click heart for me. And then that's one thing I want to ask. So this seminar already, basically released into the public for free. You can watch many video, more than 100 video. We currently have 178 pioneers, including Nobel Prize winner Frances Arnold, my academic grandma, and then 163 rising stars from all over the world. And then we already published one roadmap document, I think 3 years ago, I believe. And that's all about climate. Currently, we are writing space roadmap. Why not? My last name is Moon. My vacation next year will be in the moon. That's my plan. And then this is again, the same thing. If you are interested, you can watch by copy and paste the Zoom link online, real time. Or if you don't want to go there, late, because that's African time, probably afternoon or evening, or you can just YouTube watching. That's another thing you can do. And then, as I mentioned earlier, I have the multiple different things happen to me. One of them is discrimination, harassment. That actually happened to Tatenda Chopra. And then I realized I need to let people know about that incident to inspire. So if you are unhappy, if you think your life is too miserable, you can type my name and Science Journal. You can find one-page article, and then you may find this paper inspiring, and then make your life probably better. And then this is about your colleague Tatenda Chopra and myself. I'm chief editor of many different journals, and if you are interested in publishing, you can submit the paper to my journal, New Biotechnology. I also start another journal that is the Biocatalyst and Agricultural Biotechnology. And then I'm also chief editor of that journal as well. Plastic is one big thing about me. So I'm currently taking the paper from the community about the plastic upcycling, degradation, etc. So with that, I want to thank you again for your attention. Also, thank you for, your invitation. Also, of course, as I mentioned earlier, all the work is done by my student and my postdoc, my technician, not by me. They should get all the credit, but most of the time I'm getting credit, so I'm very, uncomfortable doing about that kind of the academic practice. But I'm again, thank you all the people working hard every day to solve the global problem passionately. And I believe they are champion. Because of them, I am very positive, optimistic, even though we have the not good world at this moment. But I believe you, young people, will change the culture by developing new solutions to the global problem. And I rely on you because you are the future. So don't be disappointed. Don't give up. Dream high and work hard. One day, you will become second Tatenda Chopra. And that is one thing I wanted to see from all of the world, and especially from Africa, because that is something we can change the current culture and current world in a better place. Once you do that, the only one benefit I can get is I probably already too old at the time, but my daughter will living in much better world because of you. So that's why I rely on you. I invest my time to you, and because I want to see that before I die, at least my daughter living in a better world. So thank you for your work, and then working hard, dream high, never give up. So I guess that's pretty much it. I see some question from chat, I believe. Thank you, Ice, posting YouTube channel, I guess. I
Yes,
do
we will
not
share
see
that.
any.
Yeah,
I do not
so
see any. Yeah.
yeah, thanks a lot, Tae Seok. Again, maybe like if anyone has like the final question to Professor Moon, you can type in, but like we probably give only maybe one or two question if it's like about inspiration or sort of, because like Professor Moon will have to leave soon for another task that we're having. But again, thank you a lot for being here with us and for inspiring us, especially for the African community. I think like they really appreciate you, specifically telling them the story, like both your personal ones and the one from your team.
Yeah. So one thing I want to tell you is don't be shy. Just send me email if you want to talk to me in person, not in person, on the virtually. I'm happy to set up a time for Zoom meeting. And then one thing I want to tell you is my wife keep telling me why you are doing so many different things, including meeting people I do not know. I'm actually meeting a lot of people every day, minimum spending 1 hour per day. Just meeting some random people I have no idea who they are. And my answer to her is, I strongly believe I'm too old to change the current world within my 20 years of, I don't know how many years I could live from now. But you will live much longer than myself. And then I talk to you, and then you will become much better leader than current leaders. And then one day, let's say 10 years from now, or 20 years from now, I watching TV, and then I see you, one of you, and then I want to say to my wife, I told you, these people become next Bill Gates, next to whatever your role model, Nobel Prize winner. And I want to say to my wife, I told you, spending 2 hours today, if I help one of you inspire from your miserable life, that's my contribution to this world. And I'll be delighted to see you in TV. You don't need to do anything for me. Just make the better world for my daughter. And that's all I ask to you. I see some question from the chat, I guess. I'm having so much problem learning things and experimentation for SynBio as it is not so expert in India. I'm too desperate to solve problem through it. So as I mentioned, if you're sick, you can find one of the good, material could be YouTube video we generating every week. You can watch that one. If it might be too difficult for you to understand at the beginning, but nobody born as Nobel Prize winner. They start with the plus minus calculation, right? And then they accumulate. And then at the end of the day, you basically mastering differential equation after many years. So you need to kind of be patient, going one step at a time. That's the key thing you need to understand. Nothing can be done overnight, but over years, I never thought I'm becoming YouTuber entire my time, 50 years, but I became YouTuber because I'm, wanted to understand young people, wanted to deliver free education. So your future, you never know. Just keep going in, and then you will see yourself become one of the leaders in the world. Tatenda never imagined he become the world hero, right? So I have another question. What is your final word over science, innovation, and business? Unfortunately, science innovation at this moment, a little bit not happening often than I expected, because people kind of rely on very incremental advance. That's kind of because of current, rewarding, practice. But again, you can dream high. You can make things change because you are the future. Your imagination is the limit, basically.
Okay, thank you so much, Prof, and thank you so much, Ice. I would like to appreciate you for Tae Seok, because you were one of the inspirations to start this platform, but I think you forgot about that. But you advised me a lot in those initial stages as I was drafting the proposal. Back to you, Ice.
Thanks a lot, Kato. So maybe the one parting thought to those of you who are listening right now or listening to recording, we are trying our best to make these accessible to all of you, especially if, those of you in Africa who may not have a lot of like SynBio education going on. So please stay in touch with our community, and we have more educational flow that we are providing. We had the beginner course in the past, and we just wrap up our intermediate course, and all the materials we were using during those courses will be published on our website, and the video, the real recording will be on YouTube. So we also inviting more people who are very active stakeholders in our SynBio community to come give talk. Like the next one in 2 weeks, we invited John Cumbers from SynBio Beta to give us some insights about like the growing community of synthetic biology. And yeah, please like stay in touch. And if you have any question about the initiative, or you want to get in touch with us, just email us at SynBio4ALL@gmail.com, and we'll be happy to help you or like work together with any initiative.
Yeah, thank you, Ice and Emma. You don't want to miss the John Combs talk. He's an amazing guy. So he will talk about much better thing than I already gave today. So you don't want to miss his talk. I want to go. Please send me the invitation to watch his talk myself. I want to watch his talk.
Of course. Thanks a lot. Thanks again, Tae Seok, for like being our guest today. He's very inspiring, and I believe like
my
everyone.
honor.
Yeah.
It's my honor to meet all the future potential leaders. One of them should be Bill Gates, I believe.
Yep.
Thank you.
All right. For the interest of time, I think I will just stop the recording, and thanks again. Have a good day, everyone.
Okay, have a good day and enjoy your weekend.